Alright, so now that we've talked about enzymes, what they are, and a little bit about how they work, now we're going to talk about how your body regulates them. Because, if you think about it, your body produces enzymes, but it doesn't necessarily make enzymes just for one reaction, and then the enzymes basically just get degraded. Sometimes you might have enzymes that are in your cell that are hanging around for a really long time, and therefore you may want to turn them on and off as needed because you don't want to make too much of particular products. So, inhibitors are things that shut enzymes down, and there are two types of inhibitors. The first kind is called a competitive inhibitor. Now, a competitive inhibitor, it's exactly what it sounds like. It competes for the active site of the enzyme. So I'm going to draw a very simple enzyme. Remember, they don't really look like this. And this is the substrate for the enzyme. And I'm going to draw another molecule that looks kind of like the substrate, but is not the substrate. And this would represent my inhibitor. So this would be what's called a competitive inhibitor because it can fit the active site of the enzyme just like the substrate. So this, if your body was to produce inhibitors, would slow the enzyme down because maybe you don't want it making too much product too quickly. In fact, what would work nicely is this inhibitor will actually slow the enzyme down a lot when there's only a little bit of substrate. But if you were starting to get way too much substrate, the inhibitor wouldn't have as much effect. What do I mean by that? Well, imagine this. You have 10 substrates and 10 inhibitors. Now, this enzyme only has about a 50% chance of actually hooking up with the substrate. And that means this enzyme is going to be very slow. On the other hand, let's say you now have 1,000 substrate molecules. This inhibitor, only 10 of these, is probably not going to have nearly as much effect because the chance of the enzyme bumping into the inhibitor is going to be very low compared to the chance of bumping into the substrate. So, one of the ways that we know an inhibitor is a competitive inhibitor is we could actually run a test and see if we add more substrate, and this is my enzyme rate, does the rate speed back up? So, in other words, this is the normal graph I showed you in the previous lecture where I add more substrate and the enzyme becomes saturated. This enzyme is working at its maximum speed. Now, if I add an inhibitor, the enzyme is going to be slowed down, so its rate should be slower. But let's say I keep adding more substrate. Does the rate speed back up? And if it does, then this was a competitive inhibitor because adding more substrate basically overcomes this type of inhibitor. The inhibitor doesn't have nearly as much effect. The second kind of inhibitor is called a non-competitive inhibitor, and this works a little bit differently. So again, here's my little enzyme. I draw them like Pac-Man. Here's my substrate, and this time the inhibitor does not bind to the active site, but it binds to another site, which is often called an allosteric site. Now what happens is, since the enzyme is a protein, when the inhibitor binds to this non-competitive or allosteric site, it changes the shape of the active site. Notice how the active site is no longer the correct shape, and this enzyme would be shut down. So in non-competitive inhibition, rather than two things competing for the active site, this literally takes enzymes sort of out of the reaction altogether. Now typically you're not going to make enough inhibitors that you're going to shut down all the enzymes, but let's just say you add 10 inhibitors, you're now going to have 10 less enzymes working. And this also means that if I add more substrate, it's not going to speed the reaction back up because it doesn't matter how many of these little purple triangles I add, this particular enzyme can't do the job, period. Adding more substrate isn't going to overcome this type of inhibition. So your body makes inhibitors naturally, but I just wanted to also note that a lot of times toxins and poisons and pesticides and even antibiotics, that's actually how they work. They're enzyme inhibitors. The way an antibiotic treats a bacterial infection, most antibiotics actually inhibit the enzymes in the bacteria that are involved in either protein synthesis or building their cell walls. And if the bacteria can't do those things, the bacteria die, and then your body can fight them off faster. And toxins like cyanide, for example, cyanide binds to certain enzymes in the electron transport chain, so you can't make ATP. So a lot of inhibitors, a lot of toxins are inhibitors. This graph, you should know this graph, again, for the test, is the same graph I showed you before, but I've added a couple more things. So this was our graph showing saturation. that if I increase the amount of substrate, this enzyme can't work any faster. It's working at its maximum rate. If I add a competitive inhibitor, notice how the competitive inhibitor, if there's not very much substrate, the inhibitor has a very big effect. The rate of this enzyme is a lot less. But if I keep adding more and more and more substrate, the inhibitor really has no effect at all, because at that point, the amount of substrate, the chance that the enzyme is going to bump into the inhibitor instead of the substrate is very low. Notice non-competitive though. If I add a little bit of non-competitive inhibitor, in this particular graph, it's showing that this non-competitive inhibitor has knocked out about half of the enzymes. So the maximum rate that this enzyme can work is cut in half. And adding more substrate, the enzyme's never going to be able to work as quick, because in this case, every enzyme that bonded to the inhibitor is shut down. So you have less active enzymes working, and the whole thing is now going to be slower. Okay, and this is a little animation. On the left, we have competitive inhibition. Notice that this enzyme's not really shut down. As long as this inhibitor is not stuck here permanently, this is just going to slow your enzymes down, because when this inhibitor falls out, it's now open to substrate. On the other hand, with non-competitive inhibition, notice that it changes the shape of the active site, and now the substrate can't bind. All right, so I want you to be aware that your body actually has a really unique way of controlling its enzymes, and it's called negative feedback. So if I have an enzyme, and my enzyme, and this is actually very simplified, breaks this substrate into product. So the substrate comes in, and it's producing this product. What am I going to do if my body is making too much? Maybe I'm not able to break this product down fast enough. I need to stop this enzyme so that I can use up this product. Well, what your body could do is make some other thing that either breaks down the enzymes or shuts them down, but your body actually has a better way. Instead of doing that, what your body does is your enzymes are designed in a lot of cases so that this product can actually be the inhibitor. So imagine this product goes back, attaches to an allosteric site, and this enzyme is shut down. So the product shuts down the enzyme, and then as this product gets used up, the enzyme would get turned back on. And so the enzymes basically regulate themselves. It's sort of like your thermostat. If you think about your thermostat, you don't think, oh gosh, I'm cold, let me go turn off the air. Oh, now I'm hot, let me go turn on the air. Your thermostat basically regulates the air. When it gets too cold, you have it set to automatically turn off and turn on. That's what your enzymes are doing. You make too much product, and your product, this actual product, will turn the enzymes off. And so it's called negative feedback or feedback inhibition. Now remember, in your case, a lot of times this is a metabolic pathway, meaning you have an enzyme here that converts this original substrate to this middle substrate. And then another enzyme converts it to this, and another enzyme converts it to this, and this, all the way down here, is your final product. This final product, hopefully, your body is using up. But let's just say that you start to make too much. The product itself goes back and stops the first enzyme. It's an inhibitor. And now the whole pathway shuts down. If you now use up the product, that will pull the inhibitors off that first enzyme, and the assembly line turns back on again. So your body is basically regulating itself. And this is another example of a real one. So this is a pathway where this particular enzyme, this enzyme number 1, converts threonine into an intermediate, a middle thing. A second enzyme converts it to the next thing, a third enzyme, a fourth enzyme, and the final product of the pathway is isoleucine. Notice, by the way, the name of this enzyme is threonine deaminase. And it doesn't matter. Just recognize that you see ACE at the end, it tells you it's an enzyme. So hopefully your cell is using up this isoleucine, but let's say that your cell's not using all of it. Now you're starting to get too much. Well, the isoleucine itself is an inhibitor. It's actually a non-competitive inhibitor of this first enzyme, threonine deaminase. Notice how the active site has changed shape, and now this enzyme doesn't work. As you use up all of the isoleucine, your body's going to say, uh-oh, I need more. It gets pulled off of the enzyme goes back to its original shape, and this pathway can start all over again. All right, and wrapping things up, kind of this is the opposite of an inhibitor. Some enzymes don't actually have the right shape on their own. They need a helper. And those helpers are called coenzymes and cofactors. So if you look at this picture, this is not an inhibitor. This is supposed to be an enzyme, but the enzyme is not in its active form. This thing is they're calling a cofactor. When it bonds with the enzyme and the cofactor together give the enzyme the correct shape. And now the enzyme is ready to react. So it's sort of the opposite of an inhibitor. And not every enzyme needs a coenzyme or a cofactor, but some enzymes do. So what is the difference between a coenzyme and a cofactor? Well, a cofactor is inorganic. Cofactors are typically elements like magnesium, iron, potassium, zinc. These basically are metal ions. Technically they're ions with charges. And these metal ions bind to particular enzymes in the body, and they help those enzymes take on the correct shape so they can do their job. Without those, if you're missing those, if you have a deficiency, then you might have a problem where that particular enzyme doesn't work properly. Another, coenzymes. So the difference between a cofactor and a coenzyme is that coenzymes are organic. So if you see the word vitamin, so vitamin A, vitamin C, vitamin B12, it's a coenzyme. If you see an element, it's a cofactor. But what they do is the same. And here's an example. The enzymes in your eyes, they need a coenzyme to work properly. In fact, one of the symptoms of a vitamin A deficiency is something called night blindness, where you actually can't see well at night. And the reason why is simply because the enzymes in your eyes are not able to work very well. They don't have the right shape without that coenzyme attaching to them. So coenzymes and cofactors, again, their job is the same. They both activate particular enzymes. And so a lot of vitamin deficiencies, rickets, beriberi, scurvy, these diseases that are vitamin deficiency or mineral deficiencies, a lot of times what it is that your body needs these coenzymes or cofactors to make a particular enzyme turn on, have the right shape, and work properly. And that's the end of our chapter.